Hi everybody, today we're going to be talking about mitosis. Mitosis is the process whereby you go from one cell to two identical daughter cells. Cells divide for a couple of different reasons. The first one is just for reproduction. Some organisms reproduce by splitting in half. There is no variation between the parent and the daughter cells. Another reason that cells divide is to grow and to develop and to differentiate. As humans, we start as a single-celled fertilized egg. We go from that to becoming an organism with trillions of cells, all of them differentiated and doing different jobs. In order to do that, we have to make more cells. The third reason why cells divide is to renew what was there or what was damaged. Think about the inside of your cheek. Every time you eat anything or drink anything or brush your teeth, you're scraping away cells. In order to make sure you don't end up with a giant hole in your cheek, we need to renew the cells that are lost with every scrape. Cells can't just divide whenever they feel like it. There are a specific series of steps that must happen before cell division can take place. 90% of the life of the cell is spent in interphase. Everything the cell does besides actually dividing is in interphase. A new cell starts its life in G1 phase, the first part of interphase. This is where the cell is carrying out its normal daily activities, growing and making new plasma membrane, organelles, cytoplasm, etc. If the cell decides to divide, it must duplicate its DNA to make two copies, one for each daughter cell. That will happen in the S or synthesis phase. This is still part of interphase. After S phase, the cell further prepares for division in G2 phase. In G2 phase, the cell is already committed to dividing and has already duplicated its DNA, but it is preparing itself so that each daughter cell will have enough stuff to survive after the cell divides. The cell will leave interphase and enter the mitotic phase, or M phase. This phase is made up of both mitosis, which is separation of the genetic material of the cell, and cytokinesis, which is separation of everything else into the two daughter cells. In eukaryotes, the DNA is not all alone in the nucleus. It is too long to be wrapped up haphazardly in the cell. If you were to lay out the DNA in one human cell, it would be over 6 feet long. The DNA has to be packaged and condensed in an organized way in order to fit inside the nucleus of the cell. DNA is associated with special proteins called histones. The DNA and histones together are called the chromatin. If you take 8 histone proteins and wind the DNA around them, it forms a nucleosome. Nucleosomes look like little beads on a string. Wrapping the nucleosomes around and around further condenses the DNA into linear segments that we call chromosomes. Therefore, chromosomes are made of chromatin, and chromatin is made up of DNA and histone proteins. Gametes, or sex cells, like sperm and eggs, only have one full set of chromosomes. N is the number of chromosomes in an organism. In humans, one full set is 23 chromosomes. Gametes are N. They are called haploid. Somatic cells are our general body cells. They are involved in doing all the work to keep us alive. We have neurons to think and remember, digestive cells to absorb nutrients, and muscles to move our bodies. Somatic cells have 2 full sets of chromosomes. One set of chromosomes is inherited from the biological mother, and one set of chromosomes was inherited from the biological father. You can see in the karyotype that for chromosome 1, we have 2 copies. For chromosome 2, we have 2 copies, and so on. However, since we inherited each chromosome from a different person, they are not identical. We call these 2 copies a homologous pair. They are 2 versions of the same chromosome. Homologous chromosomes are similar in size and structure and code for similar information. Somatic cells have 2 copies of each chromosome, so they are 2N. They are diploid. The di at the beginning refers to the fact that somatic cells have 2 copies. In humans, somatic cells have 46 chromosomes. Somatic cells divide via mitosis. They split in half and produce two identical daughter cells. Gametes are formed by meiosis. A diploid precursor cell will go through two special rounds of meiotic division to produce four non-identical daughter cells. These gametes will combine with another gamete to produce a new organism that is different from either parent and also unique from other offspring. A single chromosome is made up of a chromatid, which looks like a Good and Plenty candy with a smashed bit in the middle. The smashed bit is a region of condensed DNA called the centromere. Every time the chromosome replicates, the chromatid loses a little bit of DNA off of the ends. There is a region on the end that has extra DNA called the telomere. This area is similar to the little plastic coated portion on the ends of your shoelace. The plastic thing is there to protect against your shoelace fraying and unraveling, and the telomere is there to protect the coding portion of your DNA from being eroded during replication. Once cells have divided a number of times, the telomere gets used up and the cell starts to lose important DNA. The current theory is that this contributes to aging. Young cells and certain other cells in your body have an enzyme called telomerase that can replace the lost telomere material. This allows the cell to divide forever. Cancer cells sometimes produce telomerase to enable them to divide uncontrollably. The chromosomes are replicated during the S phase. Each arm of the chromatid is duplicated, and the two copies are held together at their centromere. These structures are called sister chromatids, and each side is identical to the other side. Even though there are two identical copies, the chromosome is still considered one chromosome. When the cell divides, each sister chromatid will go into her own daughter cell. To do this, a protein complex called a kinetochore attaches to the centromere of each chromatid. When the time comes to divide the sister chromatid, the spindle microtubules will attach to the kinetochore and pull the sister chromatids apart. The M phase, or mitotic phase of the cell cycle, consists of two phases. One is called karyokinesis. This is where we're dividing the DNA so that each cell will get an identical copy. There are 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. You can remember it as PP Mat or Professor Please Make Another Tape. However it works for you, it's fine with me. Cytokinesis is the other part of the mitotic phase, and this is where we divide everything else in the cell into 2 daughter cells. At the end of G2 in interphase, the chromosomes have been duplicated, so they're already at sister chromatids, but they haven't been condensed yet. The DNA is all spread out like a plate of spaghetti. The centrosomes are outside the nucleus and close to one another, and they have not yet begun to form the mitotic spindle. During prophase, the chromosomes begin to condense. The centrosomes begin to migrate towards opposite poles of the cell, and the microtubule mitotic spindle is formed. In prometaphase, the chromosomes fully condense, but they are still randomly spread out throughout the cell. The nuclear envelope breaks down, and now, since there's no nuclear envelope protecting the DNA, the spindle fibers can attach to the chromosomes at the kinetochore. During metaphase, the centrosomes are on opposite sides or opposite poles of the cell. They are attached to the microtubule spindle fibers, which are in turn attached on the other end to the kinetochore on each chromatid. The spindle fibers begin to shorten, resulting in a tug of war between the 2 poles. The sister chromatids are pulled one way and then the other way, until finally they form a line across the equator of the cell. This line is called the metaphase plate. In anaphase, the centromeres that were holding the sister chromatids together separate, allowing the sister chromatids to be pulled apart by the spindle fibers. Newly formed single chromosomes are pulled towards the poles. The chromosomes have reached opposite poles by telophase. We have achieved mitosis, which is the separation of the genetic material of the cell. Now we have to go through cytokinesis, which is the separation of everything else in the cell. The nuclear envelope begins to reform, and the chromosomes begin to decondense. You can see the formation of a cleavage furrow here, where it looks like there is a belt that is tightening around the middle of the cell, pinching it in two. As the plasma membrane is forced together, it will fuse, leaving two complete cells. In plant cells, it happens a little differently. There's a cell wall, which is rigid and doesn't allow for the pinching and fusing of the plasma membrane. Instead, vesicles full of cell wall materials gather at the middle of the cell. The plasma membrane of the vesicles fuse with each other, and the cell wall materials that are inside those vesicles make what's called a cell plate. As the plasma membrane extends further, the cell wall is assembled and the plasma membrane extends to the edges of the cell, separating the two cells. The cell cycle is carefully regulated. We don't want cells to decide on their own whether they're going to replicate or not. There are 3 cell cycle checkpoints. The first one is called the G1 checkpoint. This determines if the cell is going to continue through the cell cycle and divide, or enter into a holding pattern that we call G0. In G0, the cells are not actively dividing. Only a few types of cells will continue to divide throughout the life of the organism. These are mostly cells that make up your blood cells, your skin, your hair, and other epithelial linings in your body. The rest of the cells in the body are in G0. For example, neurons and muscle cells no longer divide after growth is complete. The next checkpoint is called the G2 checkpoint, and this is where the cell stops to check that everything that it needs is present before it goes into the mitotic phase. The third checkpoint is the M checkpoint, and this is where the cell stops during metaphase to make sure that the spindle fibers are correctly attached to the kinetochores. If they are not correctly attached, the cell will not continue to divide. The cell cycle is very important because if you lose control of whether your cells are dividing or not, it can lead to cancer. A cell that can divide indefinitely has lost control of the cell cycle. This is called transformation, and it leads to the formation of tumors. Benign tumors are abnormal growth of cells, but those cells have remained encapsulated at the original site. Malignant tumor is an abnormal growth that can also spread to other areas of the body. That's it for today. See you in class.